We come all the way from Vienna. Well, yeah, there's a long way to come. And he's the Medical University of Vienna molecular immunologist.
Yeah.
Yeah, so it's a really cool function. And he's here to talk about human antibody repertoires, of which Thomas has got an incredible assay and sort of the understanding of antibody responses clearly dramatically improved by some of the work that he's been doing. So I'll let him.
Yes, thank you very much. Thank you very much, Gus, for the introduction. Thank you very much for having me. And yes, I guess what we are interested in the big picture are immune responses, antibody responses, which sort of structures does our immune system recognize. And I guess that's relevant for pathogens, for bacteria, for viruses, and we have a particular interest in the human microbiome. So I guess my talk will be a little bit focused around this. Maybe just in addition to what Gus has said about introduction about me, so I always have this background slide. Originally, I'm a molecular biologist, so really hands-on pipetting, standing in the lab. But then I realized nowadays we have larger and larger data sets, and I wanted to be able to work with those data sets. So I did postdocs around bioinformatics, machine learning, and in my lab in Vienna, we're combining those two things. So on the one hand, we do assays on robots, we measure thousands of samples, and then we use these AI methods to interpret.
Is it recorded?
It is recorded,
Oh,
yeah.
yeah.
It is recorded.
Oh,
Yeah.
is it?
Yeah.
Very good.
Sorry.
And I guess from the biological questions, why did we get excited about the microbiome? So I guess you all know these numbers that we have as many bacteria as human cells on our body. And in the last couple of years, it has been appreciated that they influence many aspects of human health, how we respond to cancer treatments, the onset of autoimmune diseases. And that's not just the presence of those microbes, it's also how they modulate our immune system. And antibodies play a key role in regulating those microbiota. So we make more than 2 grams of IgA in the gut, but at the same time, we also have systemically in the blood IgG that is circulating and binding to these microbes. And we know these responses are important from animal models, but we don't really know what do they do exactly in humans. So like which species do they recognize, which antigens, which epitopes, and how do these immune responses change mechanistically between health and disease. And the major issue is that we are simply not capable of measuring so many antigens in parallel. Because we have thousands of species in our guts, thousands of bacteria, each bacterium has thousands of genes. Every gene can encode a potential antigen, a potential structure. So we would need to look at hundreds of thousands to millions of structures. And that's just not possible with conventional methods like ELISA or peptides. And what my lab does is that we have this high throughput technology where we can study hundreds of thousands of antigens in parallel. And the key idea here is that we can combine synthetic DNA libraries with phages and next-generation sequencing, which allows us to really screen hundreds of thousands of variants. So how do we do this? We start out with genetic information. So we sequence the microbiome and we know which antigens are present. But this is not limited to the microbiome. So we can also go to databases. We can pick a genome of herpes viruses. We can pick the human genome. We can basically put any protein sequence that we can encode as DNA, we can put there. Then we take these amino acid sequences that we have annotated, and we don't express them one by one, but much rather we order them as a pool. We order a synthetic DNA pool where we can get hundreds of thousands of variants at low cost. So we end up with a lot of DNA variants encoding these sequences. And then we take this DNA and clone it inside of bacteriophages in such a way that every phage makes a different protein on the surface. So we end up with a phage library rather than having all of those antigens kind of in a mixture in the gut. We have them neatly separated on the phages. And if we then add antibodies that would bind these structures in the gut, they will also bind them on the phages. And then we just need to separate the bound and the unbound phages. We wash away the unbound ones, we immunoprecipitate the bound ones, and then we kind of have the phages with the antibody-bound structures enriched. And we can detect this by DNA sequencing because every phage carries the DNA encoding the protein on the surface within itself. So if we sequence this in bulk, we will get higher read numbers. And that's the principle. This is shown here for 2 different antigens, but we can do this for hundreds of thousands of structures in parallel. And this approach is called phage display immunoprecipitation sequencing, PhIP-seq. And this has been pioneered by Steve Ellis's lab at the Broad Institute of MIT and Harvard in Boston. And they have done this for studying viruses and human self-antigens, and we have adapted it for the microbiome. And I guess, what do we do with this technology? The big idea is that in different diseases, immune-mediated diseases, we can find a distinct immune signature that we can leverage as biomarkers. So the idea is you can take a drop of blood and we can then screen it for 100,000 of antigens in parallel. One of them are these microbiota that I've been talking about, but we have now also done a library of the human proteome. So you put all 20,000 human proteins on the phages, and we also have environmental antigens like dietary antigens that people would be allergic to, for example. And obviously, what I call here microbiota is also including pathogens, bacteria, viruses that you can also all measure at once. And by doing that, the key idea is that we try to associate microbial triggers with specific diseases, find biomarkers, and what we do is more like the cancer setting, that we look at therapy response, right? You have patients that respond to a therapy, that do not respond. Can we find any signatures in their immune repertoires that would predict this? And we have broadly applied this to different diseases, right? We have some data published on how these immune repertoire change in inflammatory bowel disease, primary sclerosing cholangitis. We have also made libraries focused on viruses, right? We can take all epitopes from SARS-CoV-2 and the original SARS and common cold coronaviruses, and we have shown how they cross-react. And we also have some basic interest in linking how genetic and environment shape the immune repertoire. So basically, this is all published work. What we have not done is anything beyond these cross-sectional cohorts, right? Usually we take patients with IBD and we take healthy controls, and then we look at differences, but we can't really know if they're causal, and we don't really know the longitudinal changes. So that's something that we addressed with unpublished work. And also, everything that's shown here is done in adults aged 20 to 80 years. So we don't really know how the immune system develops. So what I will focus on today are two unpublished stories. One of them using a longitudinal cohorts where we follow adults who develop IBD, and we already have samples 10 years before disease onset, and we just describe how the immune repertoire develops over time. And the second one is about the first year of life, having pairs of mothers and infants, having samples from the child, from the mother, and just showing how they diverge in the first year of life and how that links to human health. And then I'm not sure how fast I will be with these two topics. If we have time, there's also an interest in the lab of immunogenetics, seeing how HLA shapes our immune repertoire. So if we have time, we'll also get to it. But let's start with the first one, this idea of using a pre-diagnostic cohort to kind of look at basically the immune repertoire of patients before they develop the disease. And what's conventionally done are these cross-sectional cohorts where you have patients and controls. You can compare them and you can find diagnostic biomarkers, but you don't know really anything about causality. And one way to figure that out in humans is having a sample before disease onset, right? If you have the same individuals, like a sample before onset and afterwards, and you have healthy controls before afterwards, you can see, are there any triggers? Are there any pre-diagnostic markers? And this is hard to do because many immune-mediated diseases have very low incidence rates, right? So you would need to follow hundreds of thousands of individuals. So a few of them develop a disease, and then you need to biobank those 100,000 individuals every couple of years. And if a few of those individuals develop the disease, you can go back to the biobank samples and you can look at them. And this is probably the most famous paper demonstrating this approach was linking Epstein-Barr virus, EBV, as a leading cause of MS. What they have done, they used the biobank of the US Army. Every recruit in the US Army gets a blood sample taken every year. The prevalence of MS is about 0.1%, but over, in 10 million individuals over 10 or 20 years, they had about 800 cases who developed MS during their service, and they could just go back to the sample before onset of MS and afterwards, and then they checked for EBV seropositivity, right? They did an analysis for EBV, and they saw all of those 799 individuals were negative for EBV before MS onset, and then after MS onset, all of them were positive. So this strongly suggests EBV as a cause in this. However, the issue is they already suspected EBV. They knew what they had to look for, but there are many diseases like IBD and lupus where you would not really have an idea what the trigger, what the disease could be. So what we did is we used our FIPSIC assay in this pre-diagnostic setting. This is a collaboration with Sir Mihandro and Jean-Fred Colombel from Mount Sinai in New York. And the first author is Arno Boccomje, who's currently in the Netherlands. He did a postdoc in New York. And there we got access to this US Army biobank. So the same biobank that the work on multiple sclerosis was done on. And we picked 2,000 samples from this biobank, 200 patients with Crohn's disease, 200 of ulcerative colitis, 100 healthy controls. And then we took a sample at diagnosis, 10 years before, 4 years before, 2 years before, and then we basically measure these antibody repertoires and we see how they change. And what's shown in this first plot is just the overall stability. So if you compare the three groups, Crohn's, ulcerative colitis, healthy controls, 10 years before disease onset, the stability is pretty much the same. There's no change. But already 4 years before disease onset, the immune repertoires of the healthy people are more stable, they change less. But in the Crohn's and ulcerative colitis patients, you already see a change in stability. And that also persists 2 years before disease onset. So there is something happening in the immune repertoires already before disease starts. And our method is not limited to this high-level analysis. We can really look at the exact structures, the exact epitopes, and the viruses or bacteria that are targeted. And that's what's shown in one of these plots here. So what we see here, every dot is a different structure, a different epitope. And then you have the percent of antibody binding in healthy controls and Crohn's patients. And this is 10 years before disease onset. So at first, it looks like there's not a big difference, right? If you would take any of those single dots, any of those single antigens, they would not be significantly different. Because even if they come from the same species as EBV, for example, right? These are the green dots. Every person may target a different epitope of EBV, right? When you look at the whole virus, probably 90% are seropositive, but out of those 90%, 10% may recognize one epitope, 50% recognize another one. So I guess you need to aggregate that and you need to check, is the whole species different? But one thing that you can even see here is that, for example, a subset of these patients, these yellow dots, they come from gut microbiota, flagellants. They are already overrepresented in a set of about 20% of these Crohn's patients 10 years before disease onset. So what we then do, we run statistics on this and we show, which ones of those are significant. And turns out the most significant ones are these flagellants in a subset of patients, and also EBV is different. And then we can do this at every time point, right? We can compare these immune repertoires 4 years before onset, 2 years before onset, and at the time of diagnosis. We can basically compare this to ulcerative colitis. We can compare ulcerative colitis with Crohn's disease. So it's getting too much, and I think the focus of this talk is not really IBD, so I will kind of spare you the details. But the key idea is once we pick up an interesting signal here, like EBV, we can also look in detail what is happening there. And this relates also to all of these different epitopes, right? Is there a difference in which of these EBV structures are recognized? And the interesting thing is that EBV has a lytic and a latent phase. So when it infects humans, it's lytic for a short time, it reproduces, but then it also integrates in the genome of B cells and it remains in latency, it remains dormant, and then it can get reactivated under certain conditions. And there are specific proteins associated with these phases. so that you can assign some of them to the lytic phase reactivation. And then you can also ask, how does this change longitudinally over time? And let's just focus on the first one, panel B here. This is just like BMRF1 is a protein that's required for this reactivation. So you can see if you compare, and it's not well labeled, but it's basically 10 years, 4 years, 2 years, and the diagnosis. So you can see that a lot of those proteins have no significant difference, but this one that's driving reactivation shows kind of EBV reactivation 2 years before disease onset and at time of diagnosis. That doesn't really happen in ulcerative colitis. So there's definitely this idea that EBV reactivation is associated with Crohn's onset. So what does that mean in terms of causality? Very hard to answer. It could be that those patients have an overactivation of their immune system towards harmless things, right? The immune system may have less energy to kind of contain EBV, and that's why it gets reactivated. That's one explanation. But I guess there is also this idea of EBV as a trigger by different phenomena, like kind of depending on which B cells they infect. There's some work that we did with colleagues in Germany about Epstein-Barr virus and primary sclerosing cholangitis. We have some work of them where we also look at non-Western cohorts from Madagascar and Senegal, where we also see shifts in immune response against herpesviruses. So it's a very complex topic. I'm not the right person to talk about this. I would just say there are different mechanisms, probably what we observe in MS. There's this idea of cross-reactivity, that you make antibodies against EBV and they cross-react with neural proteins in humans. It's probably not the case in all of these, but there is some general trend that EBV can transform B cells. It changes their specificity. This has been shown in lupus. So something like that is according to our data, also happening in inflammatory bowel disease. Yes. But aside of that, I think the exciting aspect is that FIPSIC really appears to be capable of kind of charting these global changes of immune repertoires over time. And we can also use this to formulate hypotheses and how you could go on and study the role of EBV with conventional ELISAs in larger groups and see how that changes. And this is something that we are doing as a part of an EU consortium that I'm coordinating. So the idea is that we also look into other diseases like lupus, rheumatoid arthritis, multiple sclerosis, long COVID, and ECFS. And there we have access to basically pre-diagnostic cohorts for these diseases. And we aim to also link this to genetics and see how that triggers these diseases. And that still leaves open the question, why do some people develop these diseases? The most extreme case is multiple sclerosis. 90 or 95% of the adult population are seropositive for EBV. They have been infected, but only 0.1% develop the disease. So it may have to do with genetics. It may have to do with the age of infection. There's like different ideas around this. But one idea that's quite striking is this idea that already infections, exposures in early life shape your immune system later on. And that's something that we addressed in this other project that I'm presenting. The kind of this idea following how does the immune repertoire develop through life. And why is that relevant? It is relevant for these immune-mediated diseases, but I guess the more common setting where this is appreciated is like allergy and asthma. So like my introduction is around this. And there's some evidence that your gut microbiome composition in infancy as a baby influences your likelihood of getting asthma later on. And the idea is that when you're born, you're more or less sterile, right? And when you're an adult, you have a certain gut microbiome composition that remains pretty stable. And there's a critical window of opportunity in the first 1 or 2 years of life where your gut is basically exposed to microbes where the composition of those microbes is settling. And if something goes wrong in that window of opportunity, if there's perturbation, this has negative effects later on. And perturbations can be, for example, exposure to antibiotics, being born by cesarean section, and also being formula fed rather than breastfed. And if infants are born naturally and they are receiving breast milk, that has beneficial effects on the microbiome, as well as like lowering the risk of atopic diseases later on. And other factors are like family size, pet farms, like this whole hygiene hypothesis, right? The idea that if you're growing up in a too sterile environment, your immune system will overreact towards harmless things later on. And obviously, the idea is that this impacts your immune system. So my analogy is that the immune system is like a little plant, right? It grows into a tree. But if you have something that pushes it out of its way early on, it will grow into a crooked tree. And the question is if we can really ever reverse this. Doesn't look like that for allergies at the moment. And I guess the big open question is, how does the immune system develop, right? How do antibody responses develop? Is there a difference between C-section, natural birth? If you administer antibiotics to a kid, if they're not breastfed, does that leave traces in antibody repertoire? And I guess this is getting even more complicated because it's not just the antibodies that the babies make themselves. They also receive antibodies from the mother through cord blood. They are breastfed where they receive IgA and IgG. And we don't even know like how efficiently are those antibodies transferred to babies? Are they enriched for specific bacteria, antigens, and how does that impact health mechanistically? And beyond that, there's this idea that you have mucosal responses in the gut and you have systemic responses in blood. So how do they relate to each other? So that's the big idea that we tried to address in this project. This is a collaboration with Sasha Zernakova from the Netherlands, from Groningen, University Medical Center Groningen, UMCG. And they have the Lifeline Next cohort, where they have recruited 1,500 mother-child pairs, and they have been collecting samples in the first year of life. So this is a cohort that's mostly focused on microbiome. So they have sequenced the microbiome composition. They have a paper now accepted that will come out soon around this. And what we did in collaboration with them is that we followed them, the mothers and the babies, in blood and serum. So for about 100 individuals, for about 100 pairs, we have samples during the pregnancy of the mother. Then we have blood samples of the baby at birth, month 3, month 12. And then we also have breast milk samples. So when we measure our antibody repertoires in this, we can first ask, how does the absolute number of bound epitopes change? And that's roughly around 1,000 epitopes that are bound. This is similar to what we see in other cohorts. It's pretty stable in pregnancy in the mother. Then it goes down after birth in the babies at month 3, and then it recovers at month 12 roughly. And in breast milk, it remains pretty stable throughout the whole time. And again, like we did for this inflammatory bowel disease, we can then also ask what are the exact bacteria, what are the exact viruses that are targeted, and what are the immune responses that change. So let's start with pregnancy. And what's shown here is just the first time point in pregnancy, so about 3 months, and then at birth about half a year later. And you have here 50 mothers with matched samples, and every mother's sample at 3 months is correlated to the sample at birth. So you can see every mom correlates to the highest level to themselves. It's a very good correlation. It's very stable. And then we can also look, is there any change in the epitopes that they are recognizing between month 3 and month 12? And every dot here is again an antigen, and then you have binding at the first time point and the second time point. And none of those are significant except for like a whooping cough vaccination that women receive in the Netherlands. And not all of them receive it. If you already have had it some years before, you don't receive it, not all of them take it. So it makes sense that it's not 100%, but rather 70%. And then this is a different visualization of this stability. Also, if you compare the other time points, right, we also have one at 28 weeks. Any way that you can compare that, you can kind of summarize the diagonal here. You have the correlation coefficient. Pregnancy, very, very stable. But if we look at the first year in life, you have same thing that we have seen here for pregnancy shown. When you correlate the mother and the infant at birth, they also correlate very nicely, right? If you have the matched baby with the mother, the correlation is pretty much perfect because it's cord blood that's transferred from the mother to the baby. So it's expected this is 100% the same. You can see siblings, twins correlate, and there are no changes in immune repertoire. If you then move on to 3 months of age, if you compare the baby at birth with 3 months later, there's already a change, right? These correlations get weaker. And if you look at the epitopes that are bound, the babies are losing some responses and they're gaining new responses, right? This whole shift here is basically degradation of antibodies that the babies have at birth. And the older they get, the more antibodies they lose. And they start making some new antibodies. This is just like, in this case, I think it's milk proteins, right? They make antibodies against milk proteins. And at 1 year of life, if you compare the sample at birth with 12 months later, there's no correlation whatsoever between the infants, right? The same individual, same baby doesn't correlate with its own antibody repertoire. And there's a complete change in immune repertoires, right? It's a bit too small to read, but for example, Epstein-Barr virus is the bright, the salmon colored one here, right? So they pretty much completely lose the seropositivity, the antibody response against EBV. They have unique antibody responses, dark red dots against gut microbiota, against flagellum, and there are many other changes that are not marked. So there's like really a great change in this immune repertoire. And if we analyze this, if we try to do a dimensionality reduction, that we kind of summarize this data, you can see that pretty much all of those time points of the mother, of breast milk samples, and of the early infant time points, they cluster together, right? You always have the blood samples and the milk samples of mothers and even of siblings and twins grouping together. So it's kind of family specific, this immune repertoire. And the one thing that's different is the month 12 of the infants that clusters differently because there are such great changes. And you remember this cohort of IBD that we have shown before, and there we had like in adults data on how much does the immune repertoire change over 6, 8, and 10 years, right? If we compare just the healthy control cohort from that set, and you can see that in the first year of life, the immune repertoire in the children is changing more than in 10 years of adulthood. And during pregnancy or breast milk lactation, it remains very stable. And then I guess the big question is, what does that mean? How does it have any impact on any health parameters? So kind of this idea, what I've mentioned earlier on, that you have perturbations, that you have difference like natural birth, C-section, breastfeeding, does it make a difference? We are slightly underpowered for doing that, unfortunately. I mean, you can see some trends. So we did test for effects of breastfeeding, of delivery mode, if they have siblings, maternal smoking, prematurity, delivery place, infant sex. And some of those come out significant. The strongest one is kind of breastfeeding. So what's shown in these two panels here, it's the same type of plots that we have seen before. And these are milk proteins from cow. And on the x-axis, you have kids that are breastfed. And on the y-axis, you have kids that are formula fed. So if kids are fed formula, they develop anti-milk antibodies already as early as 3 months of age. And the second plot D here on the side, this is at 12 months of age. And at this time point, all of the babies have received formula or cow milk. But still, the earlier exposure kind of doesn't level out. So if you're exposed earlier onto breast milk, you make higher antibody responses even later on. And the same thing is also evident for C-section. So you have kids born naturally and kids born by C-section. And you see there's like distinct groups of antigens that are massively overrepresented in the kids with C-sections. It doesn't come out as statistically significant because it's a very small number of children, right? In the Netherlands, it's only, I don't know, 10% of kids are born by C-section, and we didn't stratify the cohort for this. But if you would do this on a larger cohort, you could probably find a number of significant associations. And then there are other interesting phenomena that if the mother is smoking, also these antibodies get, I mean, this is known from other studies that you have more antibodies against RSV and like other things that are impacted in the lung. So this also gets transferred to the babies, and that's what we see. Yes. So the paper that we did there also covers a lot of other aspects that I'm not going to talk about today. One idea is kind of the stability of breast milk. So initially when we started this out, we thought, okay, there might be dynamics in early breast milk, later breast milk. Turns out breast milk is just extremely stable. Like immune responses there don't change at all, right? This is if you compare the earliest breast milk samples with the latest breast milk sample, no change. One interesting thing is that if you compare the blood of the mother and the breast milk, there seems to be some difference in which groups of antigens are transferred. Like overall, it agrees very well, but there are some antibodies against some bacteria and viruses that appear to be preferentially transferred to breast milk. No idea how that works, but it's an interesting phenomenon. And then when you correlate it to the kids, you basically see the same thing. Another thing we did is that we kind of take this data and we link it to metagenomics, because we see changes in the immune repertoire between the antibodies, and then we compare it to the presence of those microbes in the babies. And you do see if a baby has more antibodies, that is preceded by higher abundances of an associated gut bacterium. So you can basically link these immune responses to exposure in the gut. And we can also look at infectious diseases. This is one thing that we did for enteroviruses, where we kind of studied there are some antibody responses that are stable over time. There are some antibody responses that are lost, and there are some antibodies that are newly generated in the babies. And this has a lot to do with the variable regions. So there are conserved regions in these viruses. Those antibody responses are lost, and the variable responses, they are quickly newly produced in the babies. And then finally, we also have dietary antigens in there, so you can follow how does introduction of solid food around 4 months of age, how does that change immune responses? And it's quite interesting, like at 3 months of age, this is about dominated by immune responses against milk. And that at 12 months of age, there's like a massive response against wheat antigens. This, I also don't know why, but these babies, they make super strong antibodies against wheat and like barley or all types of cereals that are not detected in adults. So I guess this is also the long-term dynamics. When is this lost in childhood? We don't really know. This would be interesting to follow up on. And we do have kind of an atlas, like an idea to make all of this data accessible. It's now submitted to a journal. And I guess once this is out, this website will go live and you can kind of look up the antigens. We have everything annotated from infectious diseases. So it's really a resource for other researchers. If you're interested in norovirus, you can look up how does the immune response develop in kids and associate with other parameters. And I guess the really big potential of this would be something that we have demonstrated for the adult cohort with IBD, like kind of looking for lasting effects. How do these kids develop? How are these antibody responses linked to allergies or later on inflammatory disease? Yeah, and that's pretty much the high-level story about this infant immune repertoire. So let's maybe take a little bit of a step back and think about what else can we do with this technology, or what are the advantages? What are other people in the field of systems immunology doing? And I think nowadays there's a great interest in this PCR and TCR repertoire sequencing, right? Meaning that you take the DNA of immune cells, you check which PDJ segments are recombined, which mutations are happening, what's the clonality, what's the diversity of these repertoires. And there's beautiful work around this, how diverse these immune repertoires are, how they link to autoimmune diseases. But the big issue is still that you don't know what these receptors recognize. What are the exact antigens, the infectious diseases, the microbiota, the viruses, the self-antigens. And I like to say, in the same way that there's a lot of dark matter in our universe, there's also a lot of dark matter in our immune system where we simply don't know what it recognizes. And FIPSeq, this screening method, is a way of knowing the actual antigens and being able to associate this with diseases. Now, there are obvious limitations with them, and one of them is the general nature of working with human samples. So whatever we do, this is now an example for cancer. The idea is you want to predict who of those patients responds and who doesn't respond. So you can measure the antibody repertoire, you can look at differences, but eventually you never know if this is really causal, right? It's just associations. You don't know about causality and you don't know the mechanisms. So if you do see an increased antibody response against the microbiota in the responders, you don't know what that means. And eventually, to validate this, we need animal models, right? The idea that you can, for example, take a germ-free mouse model, or you can just take a regular model and infect it with a specific pathogen. And you can administer pathogen, you can kind of administer treatments, and you can really prove the causality. So I think there's a great potential in using this FIPSeq technology, not only in humans, but also extending it to mouse models to kind of address causality. Another more like technical aspect are also limitations of sensitivity, specificity. So compared to an ELISA, when you do an ELISA against a single protein that you have made recombinantly, FIPSeq is pretty much always going to be worse, because in FIPSeq you measure 100,000 of things in parallel and you produce those antigens in parallel, right? So you don't have the chaperones that you might have if you produce it in a cell line, a eukaryotic expression system. And also currently we are limited by DNA synthesis. So all our antigens are encoded by DNA. And the DNA that you can currently order is a maximum of 300 nucleotides. So that means you can only do about 100 amino acids of epitopes. So we split epitopes, we tile them. We have multiple peptides per protein. So we can nicely cover what's called linear epitopes, so like sequences that are close together on the amino acid strand. But if we have conformational epitopes that are further apart than 100 amino acids, we may miss them. The same way with post-translational modifications, glycosylations, lipids that are attached that we cannot detect in this setting. So overall, it's in these cases definitely inferior to other methods like ELISAs or IgA. However, the big problem is that conventional methods are inherently low throughput, right? You can only measure maybe hundreds or thousands of antigens with ELISAs, maybe 10,000 with peptide arrays. And FIPSeq allows to screen libraries of 100,000s to millions of variants. So even if you only detect a fraction, even if you only detect 10% of the antigen-antibody interactions with FIPSeq, that's still more than with these conventional methods. And you're certainly not covering the whole space, this whole dark matter of unknown things, but you're covering a larger fraction than with any other method. So it's not a perfect method, it's just the best thing that we have at the moment. And ideally, it should always be used complementary with other methods like ELISAs or PCRs. And another advantage is that this is actually rather cost efficient. You can really use it to screen large cohorts like we did. This is like another analogy I just thought of in the train. It's a little bit like this idea you only detect the tip of the iceberg with FIPSeq, right? You only get maybe 10% of the signals. But what are the alternatives, right? If you do ELISAs or peptide arrays, you only get ice cubes, right? So better the tip of the iceberg than ice cubes. And why are not more labs doing FIPSeq? Or a lot of people have been interested in setting this up, but it's quite a demanding task. So just making these libraries, doing the bioinformatics, cloning them, it's like very demanding from the lab side, right? You need to make sure that these libraries are uniform, that you don't have too many errors. Then when you produce it, when you make these phages, if you produce the phages the wrong way, it works 10 times worse. There's only one type of plastic wire that works for this. It really matters which type of magnetic beads you use for the pull down. And when you PCR amplify this, there's all sorts of weird stuff happening by working with 100,000s of variant error. And then another challenge is that it's possible to do it by hand in the lab, but there are washing steps where on the robots we pipe it up and down 30 times. So if you have to do dozens of washing steps, it's probably not really doable manually. So you need to calibrate these robots and have it running in an automated way. And when we first worked on this, we needed about 2 years to set this up. When I then moved to set up my own lab, I needed 1.5 years to set it up, even though I'd already done it before. So that's probably one of the limiting factors why there are not so many people doing this. If you have it up and running, it's quite powerful. We need small amounts of antibodies. In theory, just 3 microliters of blood. Practically, the robots have that volume. So ideally, we work with 100 microliters. We can go down to as little as 20 microliters. And yeah, we always have some internal standards. So typically, we measure about 80 samples per plate. And yeah, that's pretty much it for now. I mean, we didn't really discuss this. It's about 1 hour, right? So maybe we leave some time for questions.
Yeah. Perfect. Thanks, Tom.
Sure.
Any questions? I've got one question. If anyone's got one. Oh.
You talked about all the sort of technical difficulties of doing the library prep and how many steps there are and how fiddly it can be.
Yeah.
Like, does that introduce lots of potential for batch variation? And do you see, like, I guess what steps are the drive batch variation if you were going to like scale this up to larger cohorts?
Yeah, so I mean, the library generation, not really, because you would clone this once and then you freeze the phages and you have a stock that you can thaw and process. The problem is that you have to expand them, right? You need to grow them, you need to make more of them. And this is a step that introduces quite a large variation. And what we do is that we make one massive batch in the beginning that we have enough for 20,000 reactions. And then we freeze that as aliquots, and that reasonably, I mean, there is basically no batch variation. But if you were to repeat this in a different lab, the results turn out slightly.
I'm just online. If you just put questions in the chat. So Jeff, no, yours is more interesting. Okay, well, we'll start. We can decide afterwards. My question is, why are there like 1,000 antigens? Like what the babies respond to? Why that number? Do you have any feeling of why it isn't 10,000 or 100? My feeling, not knowing when he printed it, I thought it'd be 100,000.
Yeah, so there is a very nice review article about this, like some guys doing back of the envelope calculations. How many B cells does a person? How many can they have? So obviously you have a lot of naive B cells that could react against something. There's, I don't know, millions, billions of those. But when you look at memory B cells that have expanded and then plasma B cells that are actively producing antibodies, they arrive at a number of about 5,000 to 10,000 reactivities that you would have at a given time.
Yeah.
So why do we only detect 10 or 20% of that? I mean, we don't have the whole space of all human antigens, right? It's like even these, what I've shown here is mostly 350,000 antigens. A lot of those are gut microbiota that not all of those people have, right?
Yeah.
And for example, here we don't have all proteins of all infectious diseases. We just have the most common epitopes. So I guess even for vaccinations, we wouldn't cover everything. Then there's a fraction of, post-translationally modified things, glycans, lipids, all those conformational epitopes. So I think 10 to 20% out of the expected space is not bad. It's not perfect. And I think it will, with any other method, it will be probably harder.
So I want a sort of lay explanation.
Yeah.
Okay, Jim, now what about your question?
It's just amazing. Thank you. What would be the performance of this compared to something like AlphaFold that's trying to predict binding? There's like a whole I'm dimly aware of this whole industry of like trying to make computational prediction and binding affinities.
Yes, so I think it's a good point, but for doing that, you would need to measure, you would need to sequence the B cell repertoire of a person, right? You need to look at the B cell receptors. So that means you need PBMCs, you need blood cells. You need to sequence them, which is not too expensive. It's probably also 100 or 200 euros. But usually it's not so easy to get access to those samples in the first place. There's a lot of blood biobank. There's not really a lot of PBMCs biobank. And also the PBMCs that you take, they contain a lot of naive B cells, right? They contain a lot of B cells that don't target anything. And there's only a very small number of memory cells that actually have expanded and that make important antibodies. So the first problem would be that you just have, I mean, even if AlphaFold were to work perfectly to do this, it would be very hard to probably find a few relevant expanded binders compared to everything else that are just naive B cells. And on the practical side, there may be a day where AlphaFold can do this. At the moment, it doesn't. I mean, there are some efforts for the other way around, that you have an antigen and you try to predict an antibody that will bind it. That works better and better. It doesn't work perfectly, but kind of this prediction, I haven't really seen any study doing this on a larger scale. I think it's a great idea, but you would also need a really large training set where you know the B cell receptor and the antibody. And there are not that large training sets for this stuff.
That's a better question than one thing. Yeah.
I like to.
Yeah, okay, thank you. Mariana's got a question online. Mariana leads a cohort, a number of cohorts for sharp children. I think she's talking about Lifelines, which is a Dutch cohort.
Yeah.
And I guess it's about whether or not we're seeing a loss of this response. Well, I think we're seeing both, aren't we?
Yeah, it's both. It's definitely a loss on the one hand, right? What I said about infectious diseases like EBV, those antibodies just get degraded. The babies apparently don't get exposed so quickly, so they don't make their own responses. But they're also making new responses. I mean, I'm not sharing it again, but there was the scatter plot, and kind of one of the axes is what the mothers have, you know, that's what the babies have lost. And the other axis is what the mothers don't have and what the babies have gained. So it's both processes at the same time.
Are there any implications for when we should be doing like infant vaccination based on the time series?
Hard to say. So I guess we only have 3 time points, right? We only have birth 3 months and 12 months. So from that, we can conclude birth is not the best, right? 12 months, but you couldn't really discriminate, is it better at 3 months or 12 months? So that, I mean, it's also a problem. Like when we started this project, I said, okay, you know, vaccinations are the perfect control in this setting, right? They should have documented when our baby is vaccinated, and then we can see, do we pick up the signal in FIPSI, like which babies are not vaccinated at all. The problem is they only have this like self-reported. They don't have access to the baby's clinical, like to the health record, and this data is incomplete. So if reviewers ask for it, we may look into it, but we will only have it for a subset. It's not very accurate. So this would be also a potential to do this with a better, yeah. I mean, key issue here is linking the health record to the individuals, and we couldn't do that.
I think that was the end or something. That's it. Just wondering if you have numbers to do any kind of like preliminary look at how like HLA diversity corresponds to antibody. Like HLA corresponds to.
Yeah, that would have been the last part of the talk.
Yeah,
I can show you one slide on that.
Yeah. Oh my, I've ruined this now, haven't I? Oh God.
Being filmed doing it.
I know.
What's the narrative?
So if I share the screen, and can you move on with this slide?
Yes.
Yeah, bonus story.
Yeah, so this is actually some work with a colleague in Hungary, Matej Mansinger, that is now coming out. And the key idea is something that we did in the past, that we correlated SNP arrays with FIPSI, right? So we had 1,500 individuals, and we measured the antibody repertoire, and then we measured And we correlate the slipper rates. So this is like a Manhattan plot with only 3 skyscrapers. So there are only 3 loci in the genome that correlate with specific epitopes. Like every dot here is an epitope, like a viral or a bacterial epitope. If you were to do this with antibody concentrations, there's like more hits coming up, but really specific epitopes, it's those 3. And HLA class 2 is the main driver. And the simple reason is that for making a high affinity antibody response, it's not enough if a B cell just binds an antigen that will only give low affinity antibodies. So what needs to happen is that the B cell needs T helper cell coactivation, the T helper cell needs to release cytokines. And for the T cell to recognize an antigen of the B cell needs to internalize it, process it, and present it on MHC class 2. And there are 10,000 alleles. So if a person doesn't have an allele that can present an epitope from a certain antigen, the T cell cannot activate it, and it will not get a high affinity antibody response. And that's kind of the idea of this explains this finding. And then the work that we did with Mate is kind of this idea that not every HLA allele binds the same number of epitopes. So there's like generalist alleles that bind very broadly to different epitopes, and there are specialist alleles that can only present a very narrow set. So what we have done together is that we have used published data, or I mean, let's say the key question is, if you have a more narrow HLA promiscuity makeup or a very broad one, does that shape how many antibodies you can make? And first of all, it turns out there's quite a wide variety of promiscuity levels between specialists and general. Yeah, the promiscuity thing we may need to think about. And it's what he called it. And the interesting thing is, if you take a specialist allele, it also recognizes a specific sequence, a specific peptide, and the general list doesn't do that, right? It's like very broad. And then we correlated this with the FIPSIC data, right? So is there an association between the generalists and specialists? The interesting thing is that for pathogens, there is like your immune response against pathogens is shaped by like how many epitopes your HLA can present. For non-pathogen, it's not. And the most interesting thing is then when you take the UK Biobank and you calculate on 500,000 individuals how high is the risk for hospitalization. If you have a very broad HLA presentation, this very nicely protects against hospitalization. And you can even do this like for single diseases that you kind of show the individuals with the lowest and the highest promiscuity over the whole lifetime. There's like a massive difference and like a twofold difference in how likely they are to have STDs, herpes, viral warts, and these sort of things. So that's like ongoing effort, and I think, or if we have larger numbers of cohorts for which we have both HLA and the antibody repertoire, there's a lot more to be done.
That's super interesting.
Yeah.
Especially as the STD reporting UK Biobank is terrible. They claim like 3 people with chlamydia. Yeah, 500,000. So you sort of know that because the antibody is about 20% or whatever that's I appreciate there's not much time, but Mariana's got a question which I think is interesting about how the actual data comes out in terms of level of response. So possibly we were sort of discussing it this morning. You know, what do you actually get out of the FIPSIC? I mean, presumably you get a sort of some sort of cell, you know, count.
Yes. Yeah.
Rather than, and then you have to.
Yeah. So yeah, I mean, what you really get out of it is a p-value for each epitope. How significantly enriched is it in the single measurement? And you also get what we call a fold change. This is basically how strongly this is enriched. And how this is normalized is a discussion of